Hello everybody. We are very glad to welcome you at this second webinar. Most of you know me, and for those that don't, I'm Celine, Business Development Director at BioMeca. Today I am with you as a moderator. That mainly means to check your questions on the chat and try to answer it while Julien speaking or at the end of the webinar. So feel free to use the chat during this webinar. Enjoy this presentation of one of our so many capabilities. And now I'll pass it over to Julien.
Thank you, Celine. Welcome to the second webinar of BioMeca. Today, I will speak about electronic force microscopy, EFM, and spectroscopy, the new perspectives of analysis of cell and tissue characteristics. How these techniques are able to offer you a new perspective for your research. To begin, I will introduce myself. So my name is Julien Chlasta. I'm CEO at BioMeca, and we are specialized in biophysics in life sciences, such as cosmetic and pharmaceutic. Our innovation team is made up different parts. The first one is to develop the new innovative field. The second part is the hardware development and 3D printing to develop a new product. Over the past 3 decades, atomic force microscopy, such as IFM, as a very powerful nanotechnological imaging tool, has provided many imaging modes to analyze biological properties such as topography, elasticity, adhesion, friction, electrical properties, and magnetism of the cells, tissue, and other. So, my job today is to tell you something about the AFM and show you how IFM is a very powerful technique which can give you more information about electronic properties of biological samples. So, we will speak about electrostatic forces and then spectroscopy. Firstly, I will introduce you briefly the IFM technique and what we can do. Secondly, I will present you the EFM technique in 2 very interesting papers and the results obtained by BioMeca innovation team on hair structure treated by tensioactive ingredients. And finally, I will end this webinar by a very interesting field, the spectroscopy, project developed by BioMeca to quantify the CXAR4 protein in proteoliposomes. Before starting this presentation, I would like to introduce the different microscope techniques with this slide in order to show you what kind of studies we can carry out. The first technique, and the more common, is actually the optical microscope. On this group, we find different kinds of microscope able to study sample from the meter as tissue up to the large proteins, about 100 nanometer, with a super resolution microscope. Then we have electron microscope, which is very interesting for its high resolution power, but only for the small samples. With this technique, we can study samples from micrometers up to the angstrom, but it's a very long preparation to study biological sample. And we have the atomic force microscopy, which is very versatile techniques because you can study very large sample about the millimeter, like this fish, for instance, up to very small sample, some nanometers, like DNA, without any preparation and tissue alteration. Now, to come back to the IFM, I will show you a very simple picture. As you can see here, there is a cantilever with a spring constant registered on which we can apply a no force that allows the move of the cantilever, which is able to indent sample. And to process this signal, a laser beam hits the cantilever to reflect on the photodiode to 4 quarters, and we record the shift of the laser beam on the photodiode. The raw data obtained is a force indentation curves. As you can see in this slide, we have the different stages on this curve. The first one, with a flat curve, corresponds to the approach stage. Then we have the exponential curve, which is the indentation stage. And finally, in red, the retract curve with a little shift, which is the adhesion of the cantilever on the sample. Then we've got precisely here the contact point that represents the point where the tip touches the sample, and from this position we fit a theoretical model to convert this curve in a Young modulus representing sample stiffness, for instance. Electronic force microscope. So let's jump to the next slide. I will speak about electric mode of AFM. Dielectric and surface potential related electrical properties play a very important roles in the various biological activities and physiological processes. Electrostatic force microscopy probes ferroelectric regions of a sample surface with a conductive cantilever. An AFM image is a result of two separate scans. One scan probes the topography of the objects, while in the other, the cantilever is raised away from the surface to the region where long-range electrostatic force begins to dominate. In this electrostatic domain, the attractive and repulsive deflection of the cantilever correspond to regions of positive and negative charge on the sample surface. EFM gives users an image that couples topography with electrical properties of a nanoscale region. Now, I will show you some results extracts from papers. I'm showing a very interesting work on the electrical properties of PC12 cells compared to neurons. So, PC12 cells, representative cells of the sympathetic nervous system, and neurons, representative cells in the central nervous system. This impressive work will be helpful in a further investigation and understandings in the effect of electrical properties of biological materials. Here, the capacitance gradient and local contact potential difference, CPD, of cells body and processes of PC12 cells, neuron, or spines were investigated by EFM. The morphology of PC12 cells is shown in A here, and neurons in E. The capacitance of these cells is measured by EFM in synapse and in the picture C and J to highlight the differences in electrical signal. The fact is that the differences of these two cells, PC12 and neurons, are not due to the electric polarization properties. Here, they try to investigate the electrical properties of spines in neurons. The spines, like this, is the process of neurons growing and developing the new synapse. This process is very precious to have more nervous signal propagation. They used EFM at the nanometer level to reveal the surface topography and electric potential. And the graph shows the distribution of the capacitance gradient of the spines. The two peaks here are very closely around the main peak previously observed in the main synapse, about 20, and they demonstrate that the capacitance and dielectric properties do not play vital roles in the differences of nervous signal activities and function of the sympathetic and central nervous systems. In summary, this approach reveals the capacity of EFM to measure the capacitance of cells in physiological conditions at nanometer level. It's very important to understand that. Now, I'm showing another interesting work on the propagation of charge along individual bacteria pili. The nanoscale imaging of charge flow in protein is very crucial to understand several cellular processes, including respiration, metabolism, or photosynthesis. Here, they show the EFM can be used to directly visualize charge propagation along the pili with nanometer resolution and under temperature condition. As you can see here, in C and D, the IFM topography approach can help to differentiate flagella and pili. So the EFM can be used in physiological conditions without any alteration nor fixation by detergents. And to understand the charge propagation along the pili, charge were injected at the single point by gently touching the pili with the IFM tip. Using this approach, the propagation of the injected charges can be observed as a sharp change in the color of EFM shift, phase shift, as you can see here. In this graph, in E, we observe an incredible increase of phase shift inside the pili, revealing the propagation of the electric message along the pili. So this method is very interesting because you can directly visualize the charge flow and the distribution of a variety of biomolecules in biological systems, such as cell-to-cell electron transfers as observed in fibroblasts under physiological conditions. So let's talk about the EFM technology in the cosmetic field and how it can be used to analyze hair. The effect of static electricity in hair can really ruin your day. Indeed, it affects hair manageability and can ruin your hairstyle or your makeup. So, understanding the mechanism underneath and how to control them is crucial to create effective hair care products such as conditioners. So, conditioners are known to change hair properties in modifying the surface potential characteristics. It's interesting to understand these behaviors at the microscopic scale. In our case, the conditioner is composed of cationic surfactants. The EFM technology enables us to go deeper in the analysis of the mechanism underneath static hair. In A, here, as you can see the AFM topography on virgin hair and treated hair. The AFM approach highlights an increase of the hair surface potential. And then, in this graph, show that there is a decrease of hair potential after 3 rinsings. In conclusion, EFM, in this case, is perfectly appropriate to study the effectiveness of a product and biological samples. Now, the third part of this webinar is about AFM for spectroscopy. This technique refers to the single point measurements in which the cantilever approach and pokes into the sample and then withdraws. What can we do with this approach? There are 4 experiments that we can do. 2 using the classic parameters of the AFM, modulus measurements and adhesion measurements. I won't talk about them today. I would like to focus on 2 other techniques, which are very interesting as well. The unfolding and stretching measurements and forcing mapping. So let's get started. To characterize transmembrane protein by high-resolution imaging and localize its binding site, BioMeca provides 2 technologies: force distance curve, based atomic force microscopy, such as FD-based AFM, for high-resolution imaging, and IFM tip functionalization with a chemical group to detect the binding site of the specific protein at the nanometer resolution. Here, I will show you the CXAR4 unfolding protein, which is included in proteolipodome. They use functionalized types as biosensor are very interesting for new applications, such as the characterization of integrins, for example, and the quality control during protein manufacturing. Indeed, it gives detailed information about the orientation of the membrane protein, the number of transmembrane domains, and the number of protein per proteolipodomes. So, The chemical functionalization enables the tip selectivity and sensitivity to interact at the molecular level. In the present study, the NTA was chosen to target histidine group belonging to histag protein. Here, the IFM scans the surface of the sample through a very thin tip positioned at the free end of the flexible cantilever, which is able to move in all directions. The analysis of the cantilever deflection gives the exact path of the tip. The measurement of the interaction force is between the tip and the sample, as well as the measurements of the indentation force. So I would like you to keep in mind that this analysis of these proteins may seem easy, but in fact, the characterization of membrane proteins embedded in the lipid bilayer of liposomes or the cytoplasmic membrane is really difficult to investigate. This slide is about TXA chemokine receptor type 4, as well-known J protein coupled receptor involved in cancer cell migration, as well as in HIV infection. So, however, many other proteins in proteolipodome has been analyzed by AFM spectroscopy, and output data confirmed the interest of using this approach. As shown in this study, AFM approach provides information about the topography of the proteoliposomes with one single instrument, as in A here. And in the graph in B, we can extract some data such as the number of protein, the measurement area, and the protein orientation. And then, as you can see in C, the CXS4 complex is composed by 7 transmembrane domains highlighted by WLC model fitting and confirmed by spectroscopy. This is one example showing the power of this technology. Of course, we already have worked on different cell types. In addition, as AFM technology does not require the labeling of proteins or their denaturation, it will totally find its place in your list of quality control tests, as well as your R&D, such as protein characterization studies, for instance. I truly believe that the AFM will become a standard method. Our team is working on it. Thank you very much to be here.
As well, just maybe I have a question for you, Julien, because I can see that in your presentation. Can we use EFM on skin explants, for example, to characterize a tensioactive effect that is on skin? What kind of result we could have with the EFM?
Yes, like on cross-section after topical product applications, we focus on electric potential. So we give you different graphics before and after application associated with high resolution imaging of the cells, topography, and EFM imaging to understand the product effects.
Okay, thank you. Maybe that's it. Let me check. No, I don't have any questions. So thank you all for your attention. And I'm delighted to announce you that we'll go on with this type of event and apply the third one in the fall. We'll let you know topic and details in August. So wish you all a very good summer and see you in September. Bye and thank you. Thank you.
Thank you.